a fur ball based warm-up question for today. So first, loading in the data. And then looking at it. So just in general, we have warm-up, example 1, example 2. So this is where all the data for today is going to be. But we're looking at fur mass and month. So let's go ahead and work through the entire thing. I didn't break it up for you, but did remind you that you can use this Command, Alt, and I to insert extra chunks. So I'm gonna do it this way where I kind of like label what each chunk is by using these hash marks. So the first thing we want to do is visualize the data. So we'll do the box plot of fur mass by month. Remember, dependent, independent. The data that we're using is, I think, chapter 18. So when we look at that, we see December, June, March, September, kind of a weird order, which I will actually address in a minute. But we can see that there's the least I'll address exam questions like at the end of class. So for the box plot, we see that there is less shedding in December, higher shedding in June, more shedding in March, and then lower, but maybe equal to June, shedding in September. So I'm going to insert another chunk. So the next thing that we have to do before we do our actual ANOVA is check if our variances are equal. So check assumptions. And we do that with the Bartlett test. So again, fur mass by month, data equals chapter 18. And we see that our variances are equal because our p-value is greater than 0.05. So now we don't have to do any transformations. We can just do the ANOVA. So let's see, warm up, AOV, and make that. So AOV fur mass by month, data equals chapter 18. To view the results, we have to open it up using summary. And we see that our p-value is very small, definitely less than 0.05. So we are going to reject the null hypothesis. At least two means are different from each other, which means we have to do a Tukey test. And I'm going to print the box plot again. So the Tukey test, we do that on the ANOVA. So warm up AOV, and I'm going to make the box plot again. Or you can just copy it from above since you've already made it. And we find, let's see, so let's look at the December chunk first. So all months compared to December, we find that all months are significantly different from December. December, significantly different from these three. Let's look at the June chunk next. We find March and June are different, but September and June are not. So March and June are different, September and June are not. And then last we have September versus March, and our p-value is less than 0.05 for that. So September and March are different. So that means December is different from all of these, June and September are not different from each other, and March is different from all of them. December is the lowest, June and September are middling, and then March is the highest. So what are our results? That means that cats shed the most in March, less in June and September, and the least in December. If you wanted to take that a step farther, you could think about biologically why that might be. You could say in cooler months, cats build their coats. When it gets warm, they lose the excess fur. So it's pretty straightforward. So yes, you would lose points if you don't do the Tukey test. The Tukey test, so you could definitely argue that the only thing you need to do is make a box plot, right? Like I could look at this and be like, December is the lowest, March is the highest, June and September are like kind of similar to each other, right? But that doesn't give you any power. If you just said that, if you just like made a figure and you're like, this is the truth, like that's not statistics, right? That's just making a figure and kind of guessing. We use statistics so that we get p-values, because the p-values actually mean a probability, right? That's what p-values mean. So if your p-value is less than 0.05, that means your probability of those events occurring, if the null hypothesis is true, is less than 5%, which means it's extremely unlikely that these events are by chance, which means that there's a significant difference between them. It's the same thing with doing the Tukey test. We do the ANOVA to say that these means are not equal to each other, which we can see looking at the box plot. But when we do the Tukey test, we can officially say which ones are different. Just looking at the box plot, these aren't error bars because it's a box plot. They're maximum and minimum values, right? But just looking at this box plot, you could argue that just looking at it, you would say that these three are the same because their maximum and minimums like cross each other. So you don't know for sure until you do the Tukey test whether December is just different from these three, or if, March is different from June and March is different from September, but June and September are not significantly different. That's why you have to do the Tukey test. Okay. So let's see. I'm gonna hop into this really quick actually before we start doing the two-factor ANOVA, because we use it in the two-factor ANOVA and I think it's just kind of easiest to see right now. So as I mentioned, this isn't in the best order, right? We'd probably want it in March, June, September, December, the way that it goes in the year. But because R does everything alphabetically, it automatically put it in this order. So we're going to use a new function called factor today. And we're going to be using it right now, but we'll also be using it later today as well. So this factor function allows us to reorder a factor, or a factor in the order that we want it to be. So the general code for that is right here, excluding these little like quote kind of looking things. You might have noticed that I've kind of formatted this a little bit differently than what you've seen before, and I promise you there's a reason for that, and I'll explain it more later. But essentially the general code is you create a reordered object, so the thing you wanted, but in the order that you wanted it in. Using the factor, you select the data, the column that the data you want to reorder is in, comma, levels equals this combine, and then you type it in the order that you want it in. That means for this warm-up, that order would be like this. I created a new object just called month 2. It just has to be a different name than what it is in the original data. You can't use month because R won't recognize it when you're trying to create the new box plot. So you have to make sure it's a new name. So I just did month 2, factor, chapter 18, $month, levels, and then I put them in the order that I want them in. So March, June, September, December. Then you use this new object and you make your box plot using that. So month 2, factor, levels, concatenate or combining in March, June, September, December. If we create that, we now have a month 2 object. And when we make our box plot using our month 2 object, we get a box plot with our month in the order that we wanted it in. So now we can it's much easier for us to see that shedding decreases as the year goes on. And it's actually a little bit easier to see that June and September are similar because they're next to each other too. So we'll be using this later today in class, but it's definitely a really useful thing for making figures more understandable. Okay, so we're doing the two-factor ANOVA today. For two-factor ANOVAs, these are often called two-way ANOVAs. Same thing with the one-way ANOVA, one-factor ANOVA. Don't worry if I say it one way or the other. The factors are just different treatments. And we have a single response variable, but we're using these two treatments. And we're varying both of these factors simultaneously, so at the same time. And we're asking, does one or both affect the response variable, or do they interact with each other? So some examples might be fertilizer and light level on plant growth. So we're looking at fertilizer and light level on plant growth, and we're seeing, do fertilizer and light level interact in some way? We have temperature and salinity on fish metabolism, physical therapy regimes, and injury type on recovery. So two factors, one response variable. Some of the main features are that there's more than one factor being tested. You can actually have a multi-factor ANOVA where you have 3, 4, so on factors, but it starts getting really complicated at that point. And usually an ANOVA isn't the best test to use anymore. Yeah, so every level of both factors is represented in combination with all other levels of the other factor. So this is so like low light and high fertilizer. So think of this kind of like when we were doing probabilities, right? So we could have like light, we have highlight, medium light, low light, and then we have fertilizer, and we have low fertilizer, high fertilizer. So we have high light and low fertilizer, medium light and low fertilizer. So these bits here are the intersections between the two factors. Our response variable is continuous. That's the same thing with the one factor ANOVA. And the two explanatory variables are categorical. So these have to be categories. They can't also be continuous. The assumptions are, again, the same as from the one-way ANOVA: independent samples, randomized samples, equal variance, and normally distributed data. We are not testing for normally distributed data. Usually you would, though. But for this class right now, we're not doing that. But we will be continue testing for equal variance. There are several different outcomes of the two-factor ANOVA. One is that you have no significance. Neither factor A nor factor B seem to affect the response. The second option is that one factor is significant, but the other isn't. At this point, it's practically a one-way ANOVA. So for that example of light and fertilizer, so we might find that light is significant, but fertilizer doesn't do anything for plant growth. You might also find that both factors are significant, but there is no interaction. In this case, we call it additive. So maybe we find that highlight low fertilizer is good. See, low fertilizer is just always good. High fertilizer is bad for plants or something. Or low light is always really good for plants for some reason, so it's like extra good, but because highlight is bad, it like kind of evens out, if that makes sense. Or we can have something where we have both factors are significant, but there's also an interaction, in which case the effects are now non-additive. So maybe medium light is good at low levels, but really, really bad for growth at high fertilizer levels. And low fertilizer levels are good at low light, but really, really bad at high light. Right? So we have like a weird kind of interaction. Things don't really line up in an additive way. We have some strange things going on here where this is really bad for some reason, even though it's usually good, but in combination with high light level, it's bad for plant growth. So generally, the goal of a two-factor ANOVA is to examine the potential interaction. If we just wanted to see if a factor was significant, we would probably just do a one-factor ANOVA. The interaction is the interesting thing that we're trying to test for. So the methodology for doing the ANOVA is pretty much the same thing as a one-factor ANOVA, where we calculate sum of squares, means of squares, and then F stats. The big difference here now is that we have 3 F calcs to compare to 3 F crits. We have an F calc for factor A, factor B, and for the interaction between A and B, which means that we technically have 6 hypotheses. We have 3 conceptual and 3 practical. So for factor A, our conceptual hypothesis for factor A, our conceptual null hypothesis, is that all means by group in factor A are equal to each other. The alternative is that at least 2 means differ from each other. Practically, the way that we actually test that is comparing the means, right? The mean squares or the variance among and within. So we're looking at our variance among A versus our variance within A. Our null, just like with the one-factor ANOVA, is that among is less than or equal to within. The alternative is that among is greater than within. There's more variation between the groups than there is inside the groups. Factor B is the same thing, that all of the means are equal to each other by groups inside of factor B is the null. The alternative is that at least 2 of the means differ. The practical null and alternative are the same thing for A, except within group or factor B. The interaction is what's different this time. So now our null hypothesis for the interaction between A and B is something that there is no interaction. Because an interaction can occur in so many different ways, there's no like kind of simple like all means are equal notation for it. It's just that there is no interaction. The alternative is that there is an interaction. However, practically in the way that we test that, it's the same as with A and B, except it's the null hypothesis is that the interaction between A and B, the variance among that interaction is less than or equal to the variance within. The alternative is that the variance among the interaction is greater than the variance within. So let's look at some of these sources of variation. When we did the one factor ANOVA, we have these sum of squares terms, remember? And that's the general format for a sum of squares is you have some sort of value minus some sort of mean squared, and you're summing them up. It is a sum of squares. In our ANOVA, we have our sum of squares total, our sum of squares among, and our sum of squares within. In our two-factor ANOVA, we have our sum of squares total is equal to our sum of squares for A plus our sum of squares for B plus the sum of squares of the interaction. Sum of squares for A and B are calculated by comparing the means of all groups by factor. So sum of squares A ignores the variation in B, and sum of squares B ignores the variation in A. So I'm not going to really get super bogged down in these equations right now. But generally, the total sum of squares total, the total amount of variation in the data is looking at the variance between each value and the grand mean. So each individual point and the total mean of all of the data. The sum of squares among is looking at the variance between each group and the grand mean within, just within the data. So for A or for B. Sum of squares within compares the variance between each value and its group mean. So these definitions are actually the same as what we did for the one factor ANOVA. The equations look different though because now we're considering two factors. In these equations, little a is the number of factors, the number of groups in factor A. Little b is the number of groups in factor B, and capital N is the total sample size. Sum of squares among is looking at the variance between factor A and the grand mean. Sum of squares B is looking at the variance between factor B and the grand mean. So the mean of each group in A compared to the grand mean, the mean of each group in B compared to the grand mean. The interaction is just the remaining unexplained variance. And sorry, this is actually a typo. That should be a T. Sum of squares, the interaction for the sum of squares, I'll just write it here as well, actually. So the sum of squares for the interaction is equal to the sum of squares total minus the sum of squares A plus the sum of squares B. Wait, I think these should be also in a parenthesis. So essentially, the sum of squares interaction is just everything that isn't the sum of squares A and sum of squares B. Like with the one factor ANOVA, each of these sum of squares has its own degrees of freedom. Degrees of freedom for the total is the total sample size minus 1. Among is the number of groups minus 1. Within is the total sample minus the number of groups minus 1. Total number of samples minus the number of groups. Among is the number of groups in A minus 1. B is the number of groups in B minus 1. Interaction is degrees of freedom for sum of squares A and sum of squares B multiplied together. So again, don't get super bogged down in any of this. If the values of the factors are not related to the population means, then the sample means will be similar to one another. So the factors don't do anything, they don't change anything, they don't affect anything, meaning that the sample means will be similar to one another regardless of factor. They should only differ by amounts comparable or less than the noise or variation in the data. So comparable to the variation inside the groups. If that's the case, then mean squares A and mean squares B should be small and should be similar in value to mean squares within. And just remember that mean squares is just our sum of squares value divided by its degree of freedom. So this is just an equation for variance. If the values of factors or the interaction are related to the population mean, so the factors actually have some sort of impact on the sample means we're collecting, then the sample means will differ and the mean squares among, mean squares A or mean squares B, or the mean squares of the interaction, or more than one of those, right? So it could be A and B, or it could be A and the interaction, but B doesn't do anything. So any combination of those should be larger than mean squares within. So this kind of goes back to this. This is pretty much just a rewording of this bottom section here. So our hypotheses that we're actually testing when we do our two-factor ANOVA, we have our null and our alternatives. So for factor A, B, and the interaction, it's that the mean squares for that source is less than or equal to the mean squares within. The alternative is then that the variance for that factor or interaction is bigger than the variance within. And remember, when we technically test this, we do that using a F-calc because we're comparing variances. So when we calculate our F-calc, we do that with our mean squares within in the denominator. So mean squares, A, B, or interaction, those are also typos, my bad, would be above this mean squares within. And that's how we get our F-calc. We're comparing our F-calc to our F-crit. But remember that for our F-calcs, if it's greater than 1, that pretty much means that the MS, A, B, or the interaction, whatever one it is greater than the mean squares within. If it's less than 1, it means that our numerator is smaller than our denominator, meaning that our mean squares from our source is smaller than our variance within, our mean squares within, which would support the null hypothesis. When we do our two-factor ANOVA, we also get an ANOVA table just like we've seen with the one-factor ANOVA, except now we have a couple extra rows. We have sources, so we have factor 1 or factor A, factor B, and an interaction. We have within and we have total. For the one-factor ANOVA, it was pretty much, we didn't have these two, it was just one p-value, one F-calc, and everything else. But with the two-factor ANOVA, we actually have 3 values because we're looking at, is factor A significant? Is factor B significant? Is the interaction between them significant? So we have 3 p-values that we're going to be looking for. Again, also like the one-factor ANOVA, the ANOVA table doesn't tell us how our means differ or which means are different from each other. So again, we have to do our Tukey test to look at how means differ from each other and to look at a figure as well to determine in which direction those vary. So this example is on page 227. So we're looking at the respiration rates of marine snails, of a marine snail that lived in estuaries, that have different seawater temperatures and salinities. So we have different estuaries, different seawater temperatures, different salinities. Our response variable is the respiration rate. So respiration was quantified using milliliters of O2 consumed per hour. And here is our data. So we have 20 parts per thousand salinity, 30 parts per thousand salinity, and we have temperature. 20 degrees Celsius, 25 degrees Celsius, 30 degrees Celsius, 35 degrees Celsius. And we have a couple of measurements per group. The steps that we work through are the same as doing our one-factor ANOVA. So the first thing we want to do is look at the data, visualize it, and see what's going on. We want to check if we have equal variances. When we check for equal variances for two-factor ANOVA, we check by both factors, which I'll explain in the RStudio section. But we have to check for equal variances by both factors. If one factor is unequal, then you have to transform the response variable and try again. For this one, we find that our variances are equal, which means we don't have to do any transformations. We calculate the sum of squares, mean squares, and f-calcs and p-values if you were to be doing this by hand or in Excel. But in R, that all happens in a single step, along with completing the ANOVA table. And then we interpret the results. So this is the completed ANOVA table for the data, for these data. So we have our degrees of freedom, our sum of squares, our mean of squares, our f-calc, and our p-values. So remember, if our f-calc is greater than 1, that means that our mean squares among is bigger than our mean squares within. So mean squares, these ones here are mean squares among for factor A, factor B, and the interaction. This one here is our mean squares within. So each of these f-calcs is calculated by putting this in the numerator and then this in the denominator. All of them are greater than 1, but this one is like significantly bigger, and then this one is kind of like just a little bit bigger than 1. Looking at our p-value, we see that it is not significantly bigger than 1, meaning our mean squares for salinity is not significantly bigger than our mean squares within. So the only ones that are significantly different or have significant differences in the means is our temperature and our interaction. So salinity is not significant, but temperature changes respiration rate, and there's an interaction between temperature and salinity. Salinity by itself, though, doesn't alter respiration rate. So if we were to graph the data, this is what we get. And I've color-coded it so that it's easier to see everything too. So red is 20 parts per thousand, blue is 30 parts per thousand. So we see, and then temperature increases along the x-axis too. So we have 20 degrees Celsius, 25, 30, 35. So we can see pretty clearly that for 20 parts per thousand, as temperature increases, respiration rate increases a little bit, but not by a lot. At 30 parts per thousand salinity, though, as temperature increases, there's a pretty sharp increase in respiration rate. So it appears the respiration rate increases quickly at 30 parts per thousand salinity with temperature. At 20 parts per thousand salinity, respiration rate increases maybe a little bit slower with temperature. And then I have this little note over here. It's kind of like a fun fact, I guess, that ANOVAs are actually types of linear models. They're based on like the people who made ANOVAs based them on lines. But yeah, so this kind of crossing point can indicate to us that there is an interaction. Like if we had graphed this but didn't do any of the tests yet, didn't find that the interaction is significant, we could look at the patterns that these fall in, right? So we say that red or 20 parts per thousand is bigger than 30 parts per thousand at 20 degrees Celsius. Still bigger. And then here at this 30 degrees Celsius mark, this pattern switches. The respiration rate for 30 parts per thousand is now higher than the respiration rate for 20 parts per thousand. And that continues over here as well. So what I would kind of pull from this, if you were to write your interpretations, would be that at low temperatures, respiration rate is greater at 20 parts per thousand salinity compared to 30 parts per thousand salinity. At higher temperatures, respiration rate is higher at 30 parts per thousand compared to 20 parts per thousand. So that's how you would break down an interaction like this. Are there any questions? I know that that's a lot of information, and like I said, I kind of skipped through a lot of the like dense calculations, but I really want to focus on these interactions and how to go about interpreting them. Okay, cool. So let's go through the R markdown file. Okay. So we've got the two-factor ANOVA. Graphing the data is very similar to how we've done it before, but we're adding a couple extra things to it. So we've got box plot, our dependent variable, this little squiggly tilde, factor 1, asterisk, you have to make sure you use an asterisk, factor 2, comma, this col equals C, and then like numbers. This adds color to factor 1. I know we kind of skipped using this LAS equals 2 last week, or 2 weeks ago, I guess. But this week we definitely want to include it because the labels on the x-axis are going to be really long and they're going to cut each other out. So we want to rotate them so that we can see the entire thing. And then data equals the specific name. And that is all broken down here as well. But I want you to remember that factor 1, the one that you put first, is the one that's going to be colored. Factor 2 is going to be put on the x-axis. So that's really, really important to remember for when you're making your box plots and for when you're deciding which order that you want them in. Neither one is technically wrong, but one order may be better for seeing patterns and for not making your figure like super colorful and difficult to understand. Yeah, so this call equals C, concatenate, so you're combining different colors together, is where you state the colors that will be used in your plot. Again, the colors will be applied to factor 1. The LAS equals 2 turns your X labels vertically. And we're going to be using that factor function, like I showed you in the warm-up, to see how many categories our factors have. So that's another use of this factor function. If you do factor, data name, $, and then factor 1 or factor 2, the columns that you're looking at, it will tell you how many levels there are, how many different categories or groups there are per factor. When we test for equal variance, it's the same, actually the exact same thing that we did last week, except we have to do it twice. We have to do it by factor 1 and by factor 2. But the code for the Bartlett test is the same. If you have unequal variance by one factor but not by the other, you still need to transform your data and then retest using your transformed data, and then make your ANOVA using the transformed data. The ANOVA function is pretty much the same as last week as well, except again, we have to include our asterisk factor 2. So this is pretty much, we're testing whether the mean of our dependent variable is different by factor 1 and or factor 2. You could technically use a plus sign, but if you use a plus sign, it doesn't test for the interaction. So make sure you use this asterisk. If you have significance by either factor or the interaction, you have to use the Tukey test. So the steps that we run through for doing a two-factor ANOVA are the exact same as the one-factor ANOVA. The only difference is that we have this factor step where we want to see how many categories there are. And I'll explain a little bit more why that's important, or why that's a step you might want to do. Okay, so let's run through the first example. So looking at our data, I already see that we have two columns. We have one called age, we have age, treatment, and then heart rate. So the data below represents heart rates for subjects categorized by age group and 3 different treatment groups: placebo, control, and caffeine. Do either of these factors have an effect on heart rate? If so, what effect do they have? So does age affect heart rate? Does caffeine level or caffeine treatment affect heart rate? Is there an interaction between the two? Remember, that's the other question that we're asking. So our hypothesis, our null hypothesis, is the same for with the one-factor ANOVA. Our variance among is equal to or less than our variance within. Our alternative is that our variance among is greater than our variance within. Specifically for this problem, our null hypothesis is that the mean heart rate will not differ by age or by treatment or the interaction between the two. Our alternative is that the mean heart rate will differ by age, treatment, or the interaction between the two. So let's look at our data. So we want to graph our dependent variable, which is heart rate. And then we have two factors. We have age and treatment. So we want heart rate regardless on the y-axis. That's the same as what we always do. But now we have two choices for our x-axis. We can either put age or treatment on the x-axis. Whatever isn't on the x-axis will be labeled by color. So there's a couple of tips for choosing which should be which. So you don't want a lot of colors because it makes it really hard. It starts getting difficult to see the pattern if everything is just really colorful. Meaning that whatever's your color factor should be the one that has the fewer number of levels. And your x-axis should preferably have some sort of order. So time, amount, age, something like that, so that it reads from low to high on the x-axis, right? We saw that with our warm-up, right? We had a time increment. We had from spring till winter. So we can use the same factor function that we used before to look at the levels. And this is already typed for you. So you just need to run this. So Control Enter. Let's look at age. So factor, chapter 18, $age. So we're looking at the factor age in the data set, chapter 18. We get a printout of what that column says, but at the very bottom we have this levels, colon, and then it tells us. The levels that we have are adult, elderly, and young. So we have 3 levels. Let's look at chapter 18, treatment. So the treatment column. Looks like we have these printouts, and then we have caffeine, control, placebo. So again, we have 3 levels. So that doesn't really help us decide on the color front, because regardless, we're going to have 3 colors. So the second criteria, our x-axis should preferably have some sort of order. Well, control, placebo, and caffeine don't really have an innate order, but age does, from young, adult, elderly. So let's put those on our x-axis, which means that caffeine is going to be represented by color. That's okay. That probably just means that your screen is wider than mine, and that it's printing out a little bit differently. Like if I widen the screen all the way across and then print it, oh, mine's still like showing 1, but that's probably what it is.
Well, yours says, oh wait, 116. Mine for like adult.
Mm-hmm.
Maybe that's just why it says 1 in 16. Okay,
Okay,
maybe that's why. All right, never mind.
Yeah, that's kind of a variable on how wide your screen is and stuff. Some variation. Okay, so yeah, so caffeine will be shown by color. In the code that we're going to use this call equals, and then this combined number that we've used a couple of times, and then numbers inside of it. So these numbers are actually associated to different colors that we're going to assign to treatment 1. When we use this call equals these colors, 2 is red, 3 is green, 4 is blue, 1 is black, which is not super helpful because it kind of makes everything hard to see. But yeah, so these are pretty much the colors that we're going to use. However, I just wanted to show you this, and this is just an option for you if you want to do this. If you want, you can check out what the actual names of colors are in base R using this colors function. Again, this is totally optional. If you want to just stick to using numbers like this, that's fine. But I thought that I would also just give you this option. So if you run this colors line, R prints out all of the colors that it has stored already. And you'll notice that there are a lot of colors. So there's 657 colors. So if for some reason, if you decide that you don't want to use 2, 3, and 4, or you want to color code by something that makes more sense with the problem that you're working with, you can type these words in for your colors instead of using numbers. The only difference is that you want to make sure you put these quotes around the color. So for example, you could do call equals C, open parenthesis, quotes, red, comma, quotes, green, quotes, blue. And that would give you the exact same results as doing this 2, 3, and 4. So just another option. I'm kind of picky about colors when I actually make figures, and I like for them to match whatever the problem is that I'm doing. So I usually like to use these colors, but again, totally up to you. You don't have to do that at all. All right, so let's take a look at the box plot for this problem, making sure that we put our treatment and age in the order that we want them to be in. So copy this or type this out. I recommend actually you type it out so that you get it in your muscle memory. Ignore these little quote things around it again. There is a reason for them, and I will again explain why at the end of class. So heart rate, we're organizing by treatment first. Treatment is going to be our colored factor times age, which is going to be our x-axis factor. All equals C 2, 3, 4. LAS equals 2. That's going to turn our axis labels. Data equals chapter 18. And if I did, let's see. If I typed colors, red, oops, forget that it does that, red, green, blue. Looks like red, green, and blue give us slightly different versions of red, green, and blue. But you see that you can still do that as well. Or like pink, cyan, you know, whatever you want. Yeah. So let's look at our figure. So we've got heart rate on our y-axis. We've got age factor 2 organized on our x-axis, and we've got our colors being associated with treatment. Unfortunately, it gets cut off a little bit, but you can still tell what they are. So we have caffeine, adult, control, adult, placebo, adult. So we have adult starting off. We have caffeine, elderly, control, elderly, placebo, elderly. In the middle, caffeine, young, control, young, placebo, young. So again, remember that R does everything by alphabetical order. So caffeine, control, placebo, alphabetical order. Unfortunately, adult, elderly, young is also in alphabetical order. So caffeine is red, control is green, placebo is blue. Looking at this, we can already kind of maybe start to see a pattern going on, that adult is a little bit higher, elderly is a little bit lower, young is a little bit higher, but maybe not different from adult. But it's kind of hard to see as well. So let's use that reordering factor equation that we did for the warm-up so that we can change these levels into young, adult, elderly. Again, this reordering is also kind of optional. I prefer it, and I think it makes these figures easier to interpret. Yeah, so we'll make our new object called age 2. We're using factor to reorder it. See, chapter 18 is our data. We're selecting the column we want to reorder. We want to reorder age. The levels that we're reordering it into, we're ordering it by young, adult, elderly. So now let's make a box plot using our new reordered object. So box plot, heart rate by treatment and age 2. So treatment is our color, age 2 is on the x-axis. The colors that we're using, we're just going to do the 2, 3, and 4 again. LAS equals 2. To rotate them vertically like this, if we did them horizontally, they cover each other. Data equals chapter 18. There. Now our x-axis is organized by young, adult, elderly. So we have young, adult, elderly. Caffeine is red, control is green, placebo is blue. All right, so we will come back to this plot once we run our analyses. Step 2, same with the one factor ANOVA, we need to check for equal variances by both factors. So Bartlett test, heart rate by age, data equals chapter 18. Bartlett test, heart rate by treatment, data equals chapter 18. I didn't run either of those. I'm going to go ahead and run them both at the same time. Doing Control, Shift, Enter. So looks like by heart rate by age, our p-value is greater than 0.05. Heart rate by treatment, our p-value is also greater than 0.05. So awesome, we don't need to transform the data and we can just go straight into doing the ANOVA using our raw data. So let's set up the ANOVA again. So example 1, AOV. Using the AOV function to do the ANOVA. Heart rate is what we're doing. We're organizing it by, you can do age treatment or treatment age. It doesn't really matter. I'm going to just be consistent. I know I wrote it the opposite way up here, but since we did treatment and then age for the other ones, we'll go ahead and do it this way too. Data equals chapter 18. I also just want to say you could, if you used your new object here, so age 2 instead of age, that doesn't change anything. You get the same results. Same for here. If I did age 2, I get the same results as if I did the original age. So that's kind of personal preference again. If you use age 2, it'll keep everything in that same order you just created. If you use the original age, it won't be in that order, but the numbers will all be the same. Then we want to look at the results of our ANOVA using the summary function. We find that age is significant. And sorry, the treatment is significant and age is significant, but there is no significant interaction between the two. That's what this column is. So we have treatment, age 2, treatment versus age 2, or treatment by age 2, or interaction between treatment and age 2. Or age if you left this way. Again, it doesn't matter. So treatment, and age. Treatment and age are both significant, but the interaction is not. So let's look at our figure again. And we can kind of see that here. So the way that I like to, let's see, where is my, there it is. I don't think I can just, no. Sorry, I'm trying to do something because I think it would be helpful. Okay. Can you see this? Can you see this like screenshot?
Are you showing it right now?
Yeah, hold on. Let me try maybe a new share then. Okay, now can you see it?
Yeah.
Okay.
Yeah.
So the way that I like to visualize these and these interactions. So let's look at the caffeine across young, adult, and elderly. So caffeine, oops, why is it being like this? So caffeine, okay. So we've got caffeine for young, adult, and elderly. Let's look then at for the control. So we've got young to adult to elderly. And then we've got placebo for young to adult to elderly. So try to visualize these as if they were all stacked perfectly on top of each other. They all follow the same pattern where we have caffeine here, or sorry, young here, adult up here, elderly down here. So they all kind of follow this general shape. So if these lines would all kind of be parallel to each other, even if they're like above or below each other, that kind of signals to you that there isn't an interaction, that the effects are additive. So another way to think of that is let's look at just young. We could probably say that young maybe it has, like, if we were to ignore caffeine treatment altogether and to just average these together, we'd say the average is maybe like, I don't know, around here. Let's do that for adult. We'd say maybe the average is like also like around here-ish, ignoring caffeine treatment, just looking at the age group. And then elderly, we would see the average is maybe down here. So we haven't even done our Tukey test yet, but we can probably predict that young and adult heart rates are about the same regardless of caffeine treatment. Elderly probably has a significantly lower heart rate regardless of caffeine treatment. Okay, looking at caffeine treatments. So now we're ignoring age group. So we're just looking at the colors. We see that caffeine, so this one's a little bit trickier to ignore age group, but we'll try to just imagine this, and this averaging together. We'd say that the average between those three is maybe like here. So that's for caffeine. Let's now average for the control. The average between these green groups is maybe like here. And then average for the placebo. The placebo average is maybe around here. So regardless of age group, we see that caffeine creates a higher heart rate than control and placebo. Control has a lower heart rate, or sorry, placebo has a higher heart rate than control, but a lower heart rate than caffeine. And that the control has the lowest average heart rate altogether. we can then kind of say that these effects between age and caffeine are additive because we see these patterns regardless of caffeine and age group. That's what I was trying to get at with this general pattern, where we're saying young is always higher, young and adult are always higher than elderly, but caffeine is always also higher than placebo and control. So the same patterns hold regardless of the second factor. When we look at our ANOVA, our Tukey test results, we can see that a little bit more clearly. And we do the Tukey test the same way as the one factor ANOVA. So example, we just do it on the ANOVA object we created. The output is a little crazy because of this interaction term, but we're going to completely ignore this interaction term. So don't even bother looking at any of this. We're just going to pay attention up here. So $ treatment, that means by treatment, we see control and caffeine are significantly different from each other. Placebo and caffeine are significantly different, and placebo and control are significantly different, which we had already kind of guessed, right? Caffeine, red, is always higher than the placebo. That's the second highest caffeine level heart rate. And then the lowest is the control. Control always has the lowest heart rate. So they are all significantly different from each other. Look at age. We see that elderly and adults are significantly different. Young and adult are not significantly different. Young and elderly are significantly different. Which again, we kind of broke that down already looking at these by groups. Young and adult, that's these two groups here. So young is here, adults is here. If we ignored caffeine treatment, if we average these three together, we find that the averages are about the same between the two. Excuse me. If we look at elderly, this group over here, if we average these three together, ignoring caffeine treatment, the average would be lower than these two. So elderly is significantly lower than adult and young heart rates, which are about the same. There is no significant interaction. And when you look at the Tukey test, you'll see some of these values are smaller than 0.05. Again, just don't worry about that though. We're focusing on these results. Generally, between treatment and age, there is no interaction. Even though some pairs may be specifically like different from each other, in general, there is no interaction between them. There's no pattern going on. So, are there any patterns among the age factor? So the first part is telling us how the mean heart rate differs within the age factor. It looks like elderly heart rates are different from young and adult heart rates, which are similar to each other and higher than the elderly heart rates. Are there any patterns among the treatment factor? It looks like all treatments are different from one another. According to the graph, the highest heart rates exist among caffeine groups, followed by the placebo, then the control. Is there an interaction between the two factors? Can you tease out how the data is behaving? Excuse me. There is no interaction. The heart rates behave the same way by both factors. Okay, so let's look at example 2. This is the one that was in the PowerPoint. So we're looking at how respiration rates differ by temperature and salinity in a marine gastropod that lives in estuaries. So let's first look at the data again. So this is example 2. So we have temperature, temp, and salinity and respiration rate. careful. I know that there's question 1 uses temperature. So when we do this one where the category or the column name is actually temp, just so that they're different from each other. So let's write our hypotheses. The hypotheses are the same, remember, for all of these ANOVAs, that our variance among is less than or equal to our variance within, and that our variance among is greater than our variance within for the alternative. Okay. For this problem specifically, that means that, what is it? Temp and, let's see, neither temp nor salinity affect respiration rate, and there is no interaction between the two. Our alternative is that temp or salinity affect respiration rate, or there is an interaction between the two. So any of those can happen, temp or salinity or the interaction. So let's look at our data again. Before we look at our data, I like to look at the factors and see how many there are of each to figure out which one should be on the x-axis, which one should be the color. So again, we'll use the factor function. Chapter 18, $, we're looking at temp. Chapter 18, $, and we're looking at salinity. Those are the two factors, the two categorical factors. Okay, so we see for temp, there are 4 factors, or 4 levels, 4 groups, 4 categories, 20C, 25C, 30, and 35. So I'm going to make a little note. There are 4 levels. For salinity, we have 20 parts per thousand and 30 parts per thousand. So 2 levels. So, sorry, Minnie. So because we don't want a ton of colors on our figure, that means that we probably want to put the temperature on the x-axis and organize color to the salinities. So let's go ahead and make that plot. So resp rate by, remember, we have to be careful about the order. If we want it on the x-axis, it comes second. If we want it organized by color, it comes first. We want salinity to be the color. So salinity and temp. Colors, sorry, call equals, concatenate. We have 2 levels, so we need 2 colors. So I'm just going to 2 and 3. LAS equals 2 to flip the axis. Data equals chapter 18. And that's what we get. So luckily, we don't have to reorder anything because these start with numbers. So R is automatically organizing them in order. So 20C, 25, 30, 35, and then it's alternating between 20 parts per thousand and 30 parts per thousand. So already we kind of see a pattern going on here. I'm just going to show you really quick what it would look like if you did it the other way. That isn't technically wrong. It just might be harder to understand. So you'd need 4 colors, 2, 3, 4, and 5. So we have more colors because each color represents a salinity now, or a temperature now. So red is 20 degrees Celsius, green is 25, dark blue is 30, light blue is 35. So we can still see these patterns here, but just more colors generally makes it a little bit harder to tell. And we're organizing by 20 parts per thousand here, 30 parts per thousand here. So neither of these figures is correct or wrong. I guess they're technically both correct, but neither of them are wrong. It's just one might be easier to understand than the other. Okay, so let's check for equal variances now. So Bartlett resp rate by, let's see, I'll just be consistent. We'll do salinity first. Oh, oops. Data equals chapter 18. Greater than 0.05, so equal variances by salinity. Check by temp. Values greater than 0.05, great. We don't have to transform anything. So let's go ahead and do the ANOVA. So what is this? Example 2, AOV. In the AOV function, let's see, resp rate by salinity asterisk temp comma data equals chapter 18. Summary of example 2 AOV. Okay. So we see that salinity has a p-value greater than 0.05, so salinity is not significant. The means aren't significantly different. Temperature, p-value is less than 0.05, so temperature is significant. However, there is an interaction between salinity and temperature. So again, when you have an interaction, that means your dependent variable is not behaving the same way by both factors. There's something extra going on. The factor acts on the response variable, or the factors act on the response variable in a non-additive way. So let's look at this box plot again. And I'm just actually gonna, and let's look at both of them. Why not? Okay. So this is the original one, the one that I was gonna focus on. So again, if we imagine a line going through these, and we look at 20, 25, 30, 35, and we're comparing between 20 parts per thousand and 30 parts per thousand, we see that this 20 parts per thousand increases, the respiration rate increases as temperature increases. Okay. If we look at 30 parts per thousand, we see that respiration rate increases as temperature increases. However, it looks like it increases a lot faster than it does here. We also can look at the pattern individually at each grouping. So 20 part per thousand versus 30 parts per thousand at 20 degrees Celsius. Respiration rate is higher for 20 parts per thousand and lower for 30 parts per thousand at 20 degrees Celsius. At 25 degrees Celsius, it looks like they're about the same, but maybe 20 parts per thousand is a little bit higher than 30 parts per thousand. At 30 degrees Celsius, 20 parts per thousand is now lower. The respiration rate is lower than the 30 parts per thousand. And that pattern continues for 35 degrees Celsius. So that's what we mean there's an interaction. There's some switch, some changing in the patterns by factors. Let's look at this figure. So this is the same data organized differently. So now we have 20 degrees, 25, 30, 35. That's what the colors represent. We have 20 parts per thousand versus 30 parts per thousand. And remember that we looked at this, we saw that salinity doesn't have a significant effect. So kind of like what I did before, try to ignore all of these colors. Try to ignore all these colors. In fact, let me No, I won't actually, but ignore all of these colors. And try to imagine averaging these numbers together. The average would probably be like right here. Ignoring these colors, trying to average those together, the average would be like right here, maybe. Those averages would be about the same. So excluding temperature, salinity, heart rate, or respiration rate by salinity at 20 parts per thousand and 30 parts per thousand are about the same. They're probably about at this 4.5 range. When we look at color, though, when we look at how temperature changes with that, we start to see some differences. We now see that it increases kind of like slowly, I guess, and is a bit more at this median range when we're at 20 parts per thousand. It's a lot more stable. Respiration rate doesn't change as much. At 30 parts per thousand salinity, we see a big change from 20 degrees Celsius to 35 degrees Celsius. Again, these changes between this group and this group could indicate to me that there's an interaction, which we already found that there is from doing the ANOVA. So the last thing we want to do is do the Tukey test. So let's see, example 2 AOV. And remember, we're going to ignore this interaction because it's just really, really overwhelming, and it's often very complicated to pick out the important parts. So let's look at salinity. Oops. So when we look at salinity, we're comparing 30 parts per thousand to 20 parts per thousand. The p-value is greater than 0.05. We already knew that it wasn't significant. We got that up here. Let's look at temperature. So starting with the 20 degrees Celsius chunk, so 20 degrees Celsius versus everything else, we find that 20 degrees Celsius is significantly different than all other groups. If we look at this figure, we can kind of see that 20 degrees Celsius, that's that red one, if we averaged those two together, it'd probably be like around here, is lower than all the others. If we looked at this graph, we could see it this way. It's right here, these two. If we average these two together, the average would be like here. So 20 degrees Celsius, the respiration rate at 20 degrees Celsius, regardless of salinity, is lower than it is at all than it is at all other temperatures. Let's look at 25 degrees Celsius. 25 degrees Celsius is also significantly different from all groups, from 30, 35, and 20. So looking at this figure, 25 degrees Celsius is significantly different, significantly greater. The respiration rate is significantly greater than at 20 degrees Celsius, regardless of salinity, and is significantly lower than the respiration rate at 30 degrees Celsius or 35 degrees Celsius, regardless of salinity. You can see the same thing in this figure, because now we're just looking at the green ones. Finally, 35 degrees Celsius versus 30 degrees Celsius. This is greater than 0.05. So there is no significant difference between 35 degrees Celsius and 30 degrees Celsius in terms of the respiration rate. So that would be comparing these blues to each other, right? So these two are not significantly different. This group and this group are not significantly different from each other. So, does salinity have an effect on respiration? Explain. So, again, salinity does not. No, salinity does not change / affect respiration rate. Does temperature have an effect on respiration rate? Explain. Looking at this, yes, it does. All groups are significantly different except for this 35 degrees Celsius to 35 degrees Celsius. So respiration rate at, what was it, 20 C is the lowest, 25 C is higher, and then 30 C and 35 C are, or let's see, cause the highest respiration rate. Is there an interaction? If so, can you describe the interaction? So yes, there is an interaction. We could describe this a couple of different ways. So yes, there is. Let's see, at lower temperatures, respiration rate is higher at 20 parts per thousand compared to 30 parts per thousand. At higher temperatures, 30 parts per thousand has a higher respiration rate than at 20 parts per thousand. So at higher temperatures, respiration rate is higher at 30 parts per thousand compared to 20 parts per thousand. Okay. Are there any questions about that? It's pretty much the same thing as doing the one factor ANOVA. There's a couple extra steps in there. And the interpretation is really the trickiest part. But yeah, are there any questions? Okay, cool. So we've got another class data collection activity. This one is looking at muscle fatigue. And we're comparing, we're looking at muscle fatigue, or the time it takes for a muscle to fatigue, by muscle type and whether it is on our dominant or non-dominant side. So we're going to be testing 3 different muscle groups. The first one is going to be the forearm. We're going to then do our tricep and our calf. So when we're doing this, for your forearm, you're going to grab a tennis ball, stress ball, pair of socks, something like that. Hold it in your non-dominant arm and then repeat with your dominant arm. And you're going to use your hand and squeeze whatever it is as fast as you can. And that's really key. You can't just, squeeze. You want to really try to wear your muscle out. When you start to feel the burn, that's the point that your muscle starts to fatigue. That's what we're going to use as our metric, when it starts to burn. So you're going to time yourself with your dominant and non-dominant side to see when your muscles fatigue. We're then going to do that with triceps. So use like a can of soup, beans, something kind of heavy-ish. You're going to do tricep raises. So put your arm behind your head, lift up, and you're going to kind of do this as quick as you can until your muscle starts to burn. For calf, you're going to, hold on the back of a chair or against a wall, stand up on one leg, and do calf raises with your dominant and non-dominant foot for as long as you can until it starts to burn. Record the time, and let's do seconds for these measurements. It'll be easier to do. And we're going to collect the data and analyze and see if there is some sort of interaction between the two. So go ahead and work on that. I'm going to go ahead and make a Excel sheet and share the link for you in 1 second. So let's do file.choose, or if you're using RStudio Cloud, go ahead and upload it. It's just called the Lab 11 Muscle Fatigue 9B. So copy all of that, Control C, paste it in here, Control V. It's not hard to type. I have like such weak forearms. It's really upsetting. I've been like trying to work on that for a while. But yeah, my like left arm, forearm. And I didn't even feel it in my calves really, but my little cheeks were burning on those calf raises. All right, so looking at our data, we have our muscle group, side, and seconds. Okay, I think I got all the typos and things like that, so it should be good. Let's go ahead and do the factor thing. So muscle, $, let's look, how many categories do we have for muscle? And how many categories do we have for side? We already know, but good practice to code it out as well. So this is it just printing out everything from that column. But if we look at the bottom, oh, it looks like we have some typos, unfortunately, that I'm going to have to go through and fix for these. So I will fix that really quick. But we have calf, forearm, and tricep. So 3 levels for muscle, 2 levels for side. Let me fix the data really quick though. See, then we have 4 space. Yeah. Okay, and then oh. So unfortunately, because everything needs to be typed exactly the same, otherwise R is going to think it's a different category. We have to go through and make these adjustments. Okay, and then we also have tricep with a space. Tricep. Okay, so those typos were hopefully fixed. If you downloaded the CSV, I'm gonna re-upload it right now. Shoot. Or you could click the link and download the CSV from there as well. So file, download as a comma separated values. And then me delete the old one and upload the new one. Okay, so the new, hopefully correct one is now on Beachboard. Go ahead and download that. Now we can try this again. So now it looks like we do have just 3 levels: calf, forearm, and tricep levels, dom, and non. So I'm going to also insert a new chunk for the box plot. So we have 3 levels for muscle, 2 levels for side. Neither of them really have an order to them, so neither of them should necessarily be on the x-axis. But muscle only has 2 levels, which means there'd only be 2 colors. So let's do that one as our colored factor. So that means that seconds, which is our dependent variable, is going to come first. So we're organizing seconds by, we want the color variable coming first, so side, asterisk, then muscle. Data equals muscle with a lowercase. Sorry about that. That double muscle thing, I didn't think about that. Oh, and of course, sorry, color equals, we'll just do 2 and 3, be blue and green again. LAS equals 2. Control Enter. Okay, so our box plot is a little messy. It's a little bit hard to see because we've got these huge outliers. Which is fine, but that will make this analysis a little interesting. So let's check our assumptions. Our plot, actually, let's look at our plot a little closer. So we have seconds, we have calf, forearm, tricep organized by the x-axis, and then our colors are alternating dominant, non-dominant, dominant, non-dominant, dominant, non-dominant. So we already can kind of actually see a little bit of a pattern here where calf is maybe a little bit lower, forearm a little bit higher, and then tricep the highest maybe. And as we can already kind of see a pattern that our red is usually a little bit higher than green. So dominant is a little bit higher than non-dominant. So okay, so we plotted our data, check our assumptions. We didn't really write our hypotheses, but they would be that variance is our, that our variances among is less than or equal to our variances within for our null hypothesis. Here, I'll just type those in actually up here, just to be consistent. So our null far among or between is less than or equal to variance within. Alternative is that our variance among is greater than our variance within. For this example, that would mean that the means differ by muscle or side. Wait, sorry, do not differ by muscle or side, and there is no interaction. Our alternative is then that means do differ by muscle or side, or there is an interaction. So let's check our assumptions. Our variance is equal by both factors. So let's see, seconds by muscle, data equals muscle. lowercase m muscle. Bartlett test for seconds by side, comma, data equals lowercase muscle. Control Shift Enter to run both of those. It looks like that by muscle, our variances are not equal. By side, our variances are. But because by muscle they're not equal, we have to transform our data. So I'm going to insert a new chunk, transforming the data. I like to label my chunks personally. The data is continuous, so we're going to do a log transformation. Let's see, trans sec, because we're transforming the seconds by log. Let's see, muscle $ sec, capital S. Let's do that actually just to be consistent. And now try the Bartlett test again. Let's see, using the transform data. So trans sec by, it was not equal by muscle. So we'll do by muscle, data equals muscle. Our variances are still not equal just by a little bit, but for the sake of completing this problem, we're just going to move on with this and pretend that this almost 0.05 value is actually equal to 0.05. So doing the analysis, let's make our ANOVA. So muscle AOV, we're organizing, and we'll use the transformed data. So trans sec by, see, muscle and side. Or you could also do side, muscle, muscle side, it doesn't matter. Data equals muscle. And then summary, muscle AOV. And it looks like nothing is significant. I'm actually surprised by that. Let's try it with the non-transformed data. I'm just curious if that would change it, because it might. Nope. So it looks like trans sec. So it looks like nothing is significantly different. All of these groups are actually the same, which is pretty interesting. I'm guessing that there's a lot of these outliers are probably messing with that a little bit, with the results of that a little bit, but that's fine. So yeah, we find that there is no significant difference actually. So is there a difference in muscle fatigue by muscle? No. Is there a difference in muscle fatigue by dominant versus non-dominant? No. And is there a significant interaction between the two? No. It looks like we almost had one by muscle, but like not quite. Maybe with a larger sample size, we would have seen a difference. Yeah. Okay, cool. So that's the in class 1. Are there any questions about that? Okay. Do you want me to do one more practice problem? There's not a whole lot of time left, but I can run through one of them. If so, should I do question 1 or question 2? Some of you probably got started with these already. Okay. Yeah, I'll do question 2 then. So the black surf perch, E. jacksoni, and striped surf perch, E. lateralis, are two very similar species of fish that co-occur in rocky subtidal reefs off the northern Channel Islands. Below are the biomass of stomach contents of some males and females. So let's look at the data. So we've got question 2, biomass, species, and sex. So species and sex are two factors. So write our hypotheses. So variance among is less than or equal to variance within. Variance among is greater than or actually just greater than variance within. Specifically for this problem, that means that the means do not differ by sex or species, and there is no interaction. Our alternative is that means differ by sex or species, or there is an interaction between them. So graphing the data. Let's do the factor again to see which one should be on X, which one should be colored. So chapter 18, $, looking at species, factor, chapter 18, and sex. So it looks like there are 2 levels for both of them. 2 levels for this, 2 levels for this, and neither of them have a specific order, right? One fish doesn't come before another, male doesn't come before female, or the other way around. So we're just going to ignore that, and we can put them in whichever way we want to. So box plot, biomass. How is that spelled? Is it capitalized for both? No, just biomass. Okay. Biomass. Let's do species on the x-axis, and sex will be colored. So sex by species, because the colored one comes first, x-axis one comes second. Color equals C. I'll just do 2 and 3 again. LAS equals 2. Data equals chapter 18. Okay, so we have E. jacksoni and E. lateralis here and here. Let's check if our data meet our assumptions using the Bartlett test. We have to do it by both factors. So biomass by sex data equals chapter 18. Bartlett test, biomass by species data equals chapter 18. Sorry, there's a helicopter going overhead. Control Shift Enter to run them at the same time. And we see that by both factors, our variances are equal. So yes, don't need to transform. So we can go straight into doing the ANOVA. So Q2 AOV, let's see, biomass by sex, and using the asterisk, species, data equals chapter 18. Let's do the summary of Q2 AOV to see the results. See that there is no difference in the means by species, but there is a significant difference by sex. There is no difference. By species there is, and there is an interaction. So we need to do our pairwise analysis, and I'm going to print the box plot a second time so that we can look at it. Let's key on our Q2 AOV. underneath that, I'm going to make the box plot. And I'm actually going to make the box plot the other way as well, because it might be that will make seeing some patterns easier. So Control-Shift-Enter to run all of those together. So we see that between male and female, there is no significant difference in stomach content biomass. Oops. Okay. By species, there is a significant difference, and then we already know that there is an interaction. So we're going to ignore this again. We're just looking at these. Let's look at our figures. So this one has species on the x-axis and color is sex. It's getting cut off again, unfortunately, but we know that R does everything by alphabetical order, which means F comes before M. So this is female, this is male, this is female, this is male by color code. So looking at species, it looks like ignoring sex, that if we kind of averaged these two together, they'd probably be around here, that E. jacksoni has a lower stomach content biomass compared to E. lateralis. If we average these two together, it'd probably be up here. We also know there is an interaction though. So for E. jacksoni, we see that females have a smaller biomass for their stomach contents compared to males. But for E. lateralis, the reverse is true. Females have a higher biomass stomach content compared to E. lateralis. Let's look at our other figure, which is organized with sex on the x-axis and color is species. So it looks like red is E. jacksoni and green is E. lateralis. These are female, these are male. So we can see the exact same pattern here actually by sex. So if we kind of averaged females together, it'd be around here. If we average males together, it'd be around here. There wouldn't be a difference between the two of them. So no difference between sex. By species, so if we averaged, let's see, lateralis for males and females together, it'd be maybe like around here. If we averaged stomach content biomass for E. jacksoni for females and males, it'd be like around here. We'd see that it's lower, which is the same thing we got from this figure. But this one actually makes, I think, the interaction a little bit clearer, where we see that for males, they're the same, regardless of species. But for females, stomach content biomass differs by species. So those are our results. Is there a difference in stomach content by sex? No, that's what these results tell us, right? There is no difference by sex. If we completely ignore species, there is no difference. Is there a difference in stomach content biomass by species? Yes. Which one is higher? Looks like E. lateralis. E. lateralis has a higher stomach content biomass. Is there an interaction between them? Yes. For males, biomass is the same regardless of species. For females, let's see, what was it? E. lateralis. E. lateralis has a higher stomach content biomass compared to E. jacksoni. And that would be the interaction for this one. Okay, so there's not much time left to put you in your groups. So I'm not even going to bother. But kind of going back to why I changed the formatting of the R Markdown for this, the reason that I did that is because using R Markdown files, you can do something that's really cool called knitting. And what knitting does is it prints this out of R Markdown into either a HTML file, a PDF, or a Word document. The only thing you have to make sure you do before knitting is either delete or mark out the file.choose functions. So I'm going to use Control F to find this file.choose. And I'm going to just hash it out for all of these, or delete it, whichever one. And it looks like those are the only two. So hashing them out so that it doesn't run. And now I'm going to click on this knit button. Knit. And it takes a couple of seconds to do it. But when you do it, it knits out into these really nice and really easy to look through documents. And I've been I wanted to show you guys how to do this earlier, but I was worried it'd be a little too overwhelming at the beginning, but I think now you can it's probably not too scary. But you can knit these documents and they come out looking really nice. So the little extra kind of like tick marks around the code, when you knit them, come out as like coded sections. Putting double asterisks around text bolds it, single asterisks around text, I think italicizes. But yeah, so this is a super cool thing that R Markdown can do. And I think that I'm actually going to ask that you submit your homework assignment for this week as an R, like as a knitted file. Because it'll make it actually a lot easier on my end to grade, and it's a really good thing for you as well because knit files won't knit if there are any errors. So it's a really good way to check and make sure that you don't have any errors in your files. So for this week, please knit your file into either an HTML, a Word document, or a PDF. It doesn't really matter. And I think, yeah, if you click on this arrow, you can choose which one you knit it to. I think that knitting to an HTML is probably the easiest one, but maybe not. It's the only one that I've really done. Let's see, hopefully it'll open. Oh, it did. Cool. Yeah, so there it is as a Word document. Yeah, so that's pretty cool. This is actually a really useful thing that a lot of people are doing now when they publish their papers. They're actually typing their entire paper in R Markdown and then just knitting it and like submitting it to be published that way. So it's pretty cool. But yeah, so knit your file before submitting it to me as a Word document, PDF, or HTML. Any of those are fine. You have a homework assignment. I'll go ahead and make that available to you. That'll be due before the class period next week, even though we don't technically have class. So you still have 1 week to do it. Your group assignment is due on the 30th, and that will be our last class together before your lab exam. So if you have any questions about any of that, you can stick around for a couple of minutes and I will answer those. Otherwise, you are free to go. And I will see you, excuse me, I will see you in 2 weeks on the 30th. Have a great rest of your day and a great week.